Can you hear me okay? Great. Good afternoon, and thank you for participating in this celebration of brain science. My name is Diasynou, and I am happy and honored to be with you today. I'm excited to discuss neural circuits and how they regulate affective processing and mental wellness. There is an echo here, right? Can we? So while we're taking care of the echo so that doesn't become annoying, show of hands, who here is convinced that the brain gives rise to our ability to make decisions, learn complex skills, form social bonds, and create memories? Show me your hands. Okay, most of us, that's great. We have no trouble accepting that the brain gives rise to our cognitive abilities, our thoughts, what we collectively call the mind. We also have no trouble conceptualizing the brain as a supercomputer with multiple components, circuits that create the mind. So I ask you, when it comes to emotion, why do we hesitate to believe that emotions are generated by the brain? For example, think about this. When we are emotionally overwhelmed, we say that we have a lump in the throat. When we are excited or anxious, like I'm right now, we have butterflies in the stomach. When we are sad, we are broken-hearted. But emotions are not generated, right, in the stomach, in the heart, or in the throat. They are generated in the brain. They are constructs of the mind, and therefore the brain, based on external stimuli, physiological responses, and prior experiences. I will argue that emotions are mental states generated by networks of interconnected nerve cells in the brain. So if we accept that, then we have to accept that mental health disorders are disorders, in fact, of brain circuits. So if we can repair brain circuits, then we can repair mental health disorders. But first, in order to heal the brain, we need to understand the brain. If we wanted to understand how a supercomputer works, we would probably start pushing different buttons. Perhaps we would take off the case to look inside, maybe take out a couple of parts, right? But we cannot do that with a human brain. We cannot start pushing buttons, take off the case, right? Or start taking parts out. So that is why we use animal models, and in my lab, we use mice. With mice, we can observe their behavior, for example, to a threatening, fearful stimulus like this cat, as well as their behavior when the stimulus is no longer, oops, that was louder, that was not, it wasn't supposed to be so loud, but it got you going though. When the stimulus no longer predicts threat. Indeed, mice can suppress their defensive reactions to a fearful stimulus, like that really loud sound, upon learning that the stimulus no longer predicts threat. And that is a process we call fear extinction. And this process is compromised in persons diagnosed with post-traumatic stress disorder, or PTSD. We can also lower devices called electrodes in the mouse brain and record the electrical activity of nerve cells. And we can genetically modify specific cells, specific neurons, to make them light sensitive. This approach is called optogenetics, and it enables us to use light, which we can deliver to the brain through an optic fiber, to modulate neural activity with enhanced spatial and temporal precision. It is important that we are able to record and manipulate the activity of various brain circuits in order to understand cause and effect relationships. This understanding is necessary for the development of effective new therapeutics. So let's consider the emotion of fear. Early studies in humans and animals, including mice, showed that a brain region called the amygdala is necessary for fear. We now know that the amygdala is not the only structure involved in fear regulation. Rather, there is a whole network of brain regions that come together in order to regulate fear. And that might not be surprising given how complex fear regulation is, and also how evolutionarily significant fear regulation is. This network includes regions of the frontal cortex, including the prefrontal cortex, and of the hippocampus, about which we're going to hear in a later talk, and it's called the limbic network. The limbic network, as we currently understand it, can explain a lot of experimental and clinical observations. But unfortunately, it cannot explain a phenomenon that we call cognitive affective syndrome of the cerebellum. Now, that is a mouthful, but it is a term that was generated 25 years ago It was created about 25 years ago, and it describes symptoms of affective and cognitive dysfunction in patients with damage to the cerebellum. The cerebellum is located at the back of our brain, and despite its relatively small size, it actually contains about half the neurons, the nerve cells, in our brains. Even though neurologists knew for decades that cerebellar lesions can cause anxiety, depression, and muted emotions, these symptoms were largely ignored, primarily because the cerebellum has been traditionally considered as a motor control center. The structure and function of the cerebellum is altered in post-traumatic stress disorder, schizophrenia, and autism. Moreover, we know that the cerebellum is activated when we remember aversive memories and during fear extinction. So in the lab, given these observations, we hypothesized that the cerebellum is connected to the limbic network, and specifically to the amygdala. And we also hypothesized that this connection regulates acquired fear. We sought to test this hypothesis with a series of experiments, some of which I will show you today. And these experiments would provide fundamental understanding about the cerebellar circuitry and its role in fear processing. Now, why are these experiments important, you're going to ask? Well, they are important because in order to develop precision therapeutics, we need circuit-level resolution. Blunt manipulations of the cerebellum can have unwanted side effects, and that is actually the case with pretty much any brain region. So for example, people who take antipsychotics because they have a diagnosis of depression can develop motor symptoms that are known as tardive dyskinesia because antipsychotic medications lack circuit specificity. So in order to test the first hypothesis that the cerebellum is connected to the amygdala, we use a technique that is known as anatomical viral tracing. With this approach, we discovered anatomical connections or wires between the cerebellum and the amygdala. Here, the axons of cerebellar nerve cells that go out to the rest of the brain are labeled red, and the cells and axons that go to the amygdala are labeled green. Regions of overlap, like this one here, identify for the first time nodes of communication between these two areas. So anatomy is great, but then we wanted to test whether this connection is sufficiently strong so that when we activate the cerebellum, activity in the amygdala can actually change. So to do that, we lower electrodes in the mouse brain and we record electrical activity, an example of which you see here in the amygdala. So I'm going to show you a graph that I know is a little complicated, but I promise you we're going to walk through it together. In the top panel, you see the activity in the amygdala in response to laser light that we can shine on the projections from the cerebellum to the amygdala, because now we have genetically modified the cerebellum to be responsive to light. So we can use light that we deliver through an optic fiber to change the activity of the cerebellum. Each row here is a trial, and each black dot is an instance, it's an electrical signal that happens in the amygdala. So what you see is that when we turn on the laser pulses, the number of electrical responses in the amygdala increases. And that is quantified in the lower panel, where the responses of the amygdala per second are plotted against time. So with experiments like this, we were able to determine that the connection between the amygdala and the cerebellum is strong enough so that when we activate the cerebellum, we can actually change the way that the amygdala responds. To test the second hypothesis that the novel cerebellar amygdala connection regulates learned fear, we actually performed behavioral experiments using a protocol that is known as associative fear conditioning. The first thing that we needed to do was to plant a fearful memory in the mouse brain. So we did that by exposing the mouse to paired presentations of a tone and a mild electric shock. The mouse is not harmed by the electric shock, but the experience is sufficiently adversive so that the mouse associates the two stimuli, and now it exhibits a defensive response in the form of freezing to subsequent presentations of the tone alone. Now, for mice, freezing is a natural and adaptive behavior whenever they are faced with an inescapable threat, and it is accompanied by increased physiological reactions such as defecation and heart rate, similar to fear responses in humans. So I'm going to show you clips that show an example of a mouse responding to the tone before and 48 hours after training. So when the clip starts, the onset of the tone will be indicated with a cartoon loudspeaker. I hope it is clear that after training, the mouse freezes more to the tone than before, indicating that a fearful memory has been successfully generated. Now, we will learn more about memory formation and maintenance by our next speaker after the break. For our purposes, we can tell by this increase in freezing that the fearful memory has been planted successfully. So now we can test the role of the cerebellar amygdala connection in fear extinction. So trained mice are split in two groups, a control group that does not respond to light, and an experimental group that has been previously engineered genetically so that the cerebellar amygdala connection can be silenced with light. In the behavioral chamber, the mouse is presented without the shock several times, and at the same time, a laser light is delivered to the brain through the optic fiber. In the control group, which is insensitive to laser light, repeated exposure of the tone alone results in gradual fear extinction, as you can see from the decrease in freezing. The question is, what will happen to the experimental group, in which the cerebellar amygdala connection is silenced? So what we find is that extinction actually happens faster. So you see that with training, both groups reach the same steady state, but the silenced group gets there faster. So I'll show you these two clips. On the left is a mouse that belongs to the control group, where the cerebellar amygdala connection is intact, and on the right is a mouse where the cerebellar amygdala connection can be silenced with light. Again, when the tone goes on, a loudspeaker is going to appear, and now a blue square is also going to appear when the laser is on. So as you can see, the experimental mouse shows significantly less freezing to the tone, so faster extinction, compared to the control mouse, which remains frozen. Importantly, this effect is not due to altered locomotion in response to light. So these results suggest that the cerebellar amygdala connection is involved in the maintenance of learned fear. Our findings agree with the observation that the cerebellum is hyperactive in people diagnosed with PTSD and suggest that silencing the cerebellar amygdala connection may be an effective treatment in fear disorders. For us, immediate next steps involve examining the dynamics of the signal in the cerebellar amygdala pathway using fluorescence sensors and real-time imaging of neural activity, as well as probing the functional specificity of the circuit. It is important that we know when the circuit is active and also whether it is involved in learned fear specifically or also in other emotions. because this way we can design successful therapeutic interventions. We are also probing the pathology of this circuit in autism. So at present, manipulation of cerebellar activity in depression, PTSD, schizophrenia, and social emotional dysfunction in spinocerebellar ataxia is under investigation, and it can be achieved in humans non-invasively with magnetic or direct current stimulation. The issue with these approaches is that they do not offer circuit specificity because they target all nerve cells in the region. It perhaps, therefore, is not surprising that their efficacy is low and that only some groups of patients respond to these treatments. So we are excited with the prospect of future technological innovations with improved specificity. But what about right now? If our advances in circuit neuroscience help us accept that emotions come from our brains, then maybe we can fight the stigma of mental health disorders and improve the lives of people who need mental health support and of everyone else around them. And with that, I would like to thank current lab members, funding sources who have supported this research, and you for your attention. Thank you.
Questions?
Thank you for your presentation. Do you use certain medications or chemicals to change what's happening in the cerebellum?
So we don't have a way to easily introduce medication in the cerebellum other than through surgery. And even in that case, where we could locally infuse a medication in the cerebellum, we have no way of controlling which circuits or which nerve cells are going to be affected. And that is the challenge with any pharmacological approach, actually. So that's why we're using these mouse models and we resort to genetic tools to try and get this circuit specificity in our manipulations.
Thank you.
Thank you very much for the presentation. I think I misunderstood you, but I didn't understand, how does one learn fear?
How does one learn feel?
Yes.
Fear.
Fear.
Well, so let's take the case of post-traumatic stress disorder, for example. An individual is experienced to a really strongly negative event, an event that causes trauma, that causes intense fear. So subsequently, when they are removed from the dangerous situation, from the fearful situation, they remain fearful of particular stimuli, for example, sounds or smells that remind them of that situation. So that is a case of acquired fear. Before the event, they were not afraid of the particular sound or the particular smell, but after the fearful event, they are. So that's how you can acquire fear versus what we would call an innate fear, which might be fear of snakes if you are a human, or fear of a cat if you are a mouse.
Does blocking the fear response to shock also eliminate other fear responses?
Blocking the fear response to
To the shock.
the shock.
Does it also eliminate other fear responses? For example, if the rat was exposed to a cat.
It shouldn't, but that's a good idea. That's an experiment we haven't done. It should be stimulus specific.
Thank you.
Thank you. You mentioned earlier that there were a variety of things that happened regarding emotion. And I was wondering if you had any thoughts on the polyvagal theory as a way to organize understanding around emotions.
Sorry, I cannot hear you. On the
Polyvagal theory?
for the vagal nerve stimulation.
Right, right.
I'm not sure Since we don't work with humans, I'm not sure, other than what is in textbooks and in the literature, in the lab, we don't have personal experience with that. It is true that in general, neuromodulation or stimulation of either the brain directly or cranial nerves, like in this case, the vagus nerve, can actually modulate emotion, but I cannot give you any inside information as to whether this works or not. Are there individuals at the Mind or CMB maybe who do vagal nerve stimulation? I don't know, but we can look that up if this is something that is of interest to you.
Hi, thank you. I understand how this research can be applied to acquired fears, like in the case of PTSD. Can you explain how it can be applied to something people are born with, like autism?
The idea that you can modulate those projections. So autism is a multifaceted neurodevelopmental disorder. And one of the things that is affected in autism is the emotional response, the emotional reactivity. And anybody who has interacted with young children with autism knows about temper tantrums and the inability to control one's emotions. So what we're trying to understand is exactly what are the pathways that might be dysfunctioning in those situations in order for this deregulation of fear to happen. Our hypothesis is that the cerebellar amygdala pathway might be one of those. So in the lab, the way we're addressing this issue is we have mice that carry mutations that humans with risk or humans that express different genetically inherited forms of autism. We have this gene or this mutation in the mouse, and now we're trying to understand what the neurophysiology of the particular pathway is.
Hi, thanks. I was wondering, is there any effect of silencing the cerebellum independent of exposure? Meaning, when you're the first trial of exposure, do they already have diminished responses relative to the control group?
Yeah, that is interesting. No, they don't. So it is not an effect on the strength of the memory or the recall of the fear. It happens subsequently. The effect is after that. There is one more. Oh, yeah.
So you mentioned mostly about the brain and the other parts of the body where we associate other feelings. You said may or may not be. How about the gut? I mean, aren't there a lot of neurons and activity there?
Right. So again, this is not something that we study, but at the Center for Neuroscience, Dr. Jennifer Whistler is very much interested in the interaction between the gut and the gut microbiome and the brain. Her research is in the context of addiction, but this is definitely something that we're looking at. You're absolutely right. Talking about brain and body interactions, our last speaker, Dr. Audrey Fahn, is also going to touch upon that issue. I think there was, one more question over there.
With disorders like amnesia and like memory loss, do you think that would affect learned fears, or would it still be like a stress response?
It depends. We know that how fear and in general how emotion is processed and expressed has to do with the internal state. It depends on what type of amnesia or what type of memory loss you're talking about, if it's specific. So it is a very complicated question what you're asking. We can chat about it later if you want. Sorry, Jennifer, we have you running around.
Hi, I was just very curious to know what would you anticipate the non-invasive stimulation techniques would look like the technology, what would that look like for your study?
For the mouse or in general for the cerebellum, for the human?
For the human.
So I would say deep brain stimulation already qualifies as invasive, right? It involves surgery, it involves implantation of electrodes. And these are approaches that already happen in humans, especially like Parkinson's patients or patients with epilepsy that is not responsive to medication or other technologies. This is not something that is currently done in the cerebellum, but I think it's just a matter of time. The limitation with invasive technologies, at least in humans, the way it's done right now, is whatever neurons are around the electrode are going to be activated or silenced. So again, you don't get this circuit specificity that we can get with those tools, but I think technology will eventually get us there.
Last question.
Would there be any other side effects of silencing the cerebellum?
Yeah, so that's a great question. So like I said, the cerebellum is traditionally considered a motor control center. So if you bluntly silence the cerebellum, the first thing that you see is ataxia, incoordination. You know how when I drink and a policeman, right, a policeman or a police person stops me and they're like, walk a straight line, show me that you haven't been drinking, and I cannot walk that straight line, that's because the first nerve cells in the brain that are affected by alcohol are nerve cells in the cerebellum. So whenever you bluntly mess with the cerebellum, that's what happens, ataxia happens. So that's why it's so important to be able to develop technologies that target specific circuits, specific connections, as opposed to the whole brain region.
Thank you so much, Dr.
Thank you very much.
Diasynou, for your own time.